afternoon, Whitney. How are you feeling today?
I'm okay, but I'm a little nervous to hear about my results. I don't know why I had to come in.
Well, Whitney, I have some very sensitive news that I have to deliver to you that I didn't think that it was appropriate to deliver over the phone. You have the gene that carries Alzheimer's. This does not mean that you will get Alzheimer's, but it does mean that you are a carrier for it.
What? I don't know how you can be so calm about telling me this. What does this mean? What causes this?
This is the hardest news for a doctor to deliver, but we don't know exactly what the reason is for Alzheimer's to develop in a brain. What I can tell you is that if the enzyme ClpXP is inactive, then it could be a cause for Alzheimer's.
Okay, you should tell me some more.
Okay, so ClpXP is an ATP-dependent protease composed of two enzymes, ClpX and ClpP. ClpX is a hexamer and AAA plus ATPase that identifies unwanted peptide sequences in the proteins before unfolding them. ClpX does this by binding to short, unstructured peptide sequences, which are called degradons, degradation tags, or recognition signals. The peptide is then moved to the proteolytic chamber of ClpP. ClpP is a 7-unit serine peptidase. It binds to ATPase and degrades misfolded proteins. ClpXP can only perform its function when there is ATP in the mitochondria. ATP is not created when the cell is under oxidative stress. Oxidative stress is when there's an imbalance, between reactive oxidative species and antioxidants. Reactive oxidative species, or ROS, that's what I'm going to call them from here on out, just to let you know, are free radicals like peroxides. There are several ways cells can be exposed to ROSs. Extracellularly, like food additives, drugs, the exposure, and UV light. And intracellularly, by processes derived by the mitochondria. The ROS prevents the production of ATP mitochondria by hindering the active sites and allowing molecules to perform their processes in oxidative phosphorylation. Therefore, if decreased ATP due to oxidative stress, then ClpXP cannot perform its function of cleaving. When ClpXP cannot cleave due to the lack of ATP, then the damaged or unwanted proteins can aggregate and cause diseases or malfunctions in the cells. This can ultimately affect intracellular pathways and contribute to disorders and diseases like Alzheimer's, diabetes, and other genetic diseases. The mechanism in which ClpXP locates and degrades substrates is well understood. ClpXP unfolds and transports the substrate through repeated cycles of ATP hydrolysis. ClpX then translocates the unfolded protein into ClpP, and ClpP degrades the unwanted sequence at either the C terminus or the N terminus, because the peptide has a C terminus or an N terminus. I don't know if you're familiar with those terms. Yet, what is unknown is how to keep the domains ClpP and ClpX stable to be able to perform research on them.
Okay, can you show me this beast that may cause me to have a very sad neurodegenerative disease one day? I mean, I just don't understand this. Neither of my grandparents had Alzheimer's disease, and my parents are not showing signs. How could I have this?
So, weirdly enough, I've brought some materials that will help you visualize this. So this, as I said before, just because you have the gene for Alzheimer's disease does not necessarily mean you're going to get Alzheimer's disease. And it's very good that you're having this test run right now. But I do have a picture, like I said, of ClpX handy. But I do not have a picture of ClpXP. So this is ClpX, and as you can see, the 6 hexamers that it's made of. This is from research that a colleague was doing of mine, and it's from an E. coli cell. And you may be wondering what the point of me showing you this is, but in an so the difference between an E. coli cell and a human cell is just the types of protein sequences that they unfold and cleave. That's the only difference. So it's essentially the same structure, but those are the only real difference.
Okay, can you give me some facts or figures, help me better understand the importance of this protease?
I thought you might ask that question too. So I brought a figure that I can show you with some other recent research that was done on Shmonella onedensis. Not very good at pronouncing things, so my apologies. But S. odenesis is a bacteria that can reduce metals aerobically and anaerobically. And this was used to see if ClpXP is necessary for degradation of misfolded proteins that occur in high concentrations of iron in anaerobic conditions. The researchers used optical density, so this is the OD on the diagram, to determine the cell growth and metabolic activities of the cell. In this figure, the y-axis is labeled OD 600, so optical density, and they're looking at wavelength 600 nanometers. And then the y-axis is labeled in time hours, which to just show the rate of the cell growth over a 24-hour period, as you can tell, 24 hours. So 2 experiments, which are the closed circles, were the rates of the growth in anaerobic without oxygen conditions in lysogeny broth, which is LB, composed of 40 millimolar fumarate and 20 millimolar lactate, and then 2.5 millimolar of iron chloride in the S anadenesis. The wild type, the squares, and RPO mu and circle. RPOE, I misspoke before, RPOE, encodes for sigma E. And sigma E is very important because it regulates for stress response in your body. And that is also regulated by ClpXP. And so that's why this whole experiment was done. And the 2 experiments that were run, the open symbols, were the rate growth and anaerobic lysogeny broth composed of 40 millimolar fumarate, 20 millimolar lactate, and then S anadenesis. The same things, wild type was the square right here, and then the circle was the RPOE mutant right here. The control used was the wild type closed square right here. This is because it is not yet mutated, but does have high concentration of iron chloride. Therefore, the author had to compare the mutated RPOE iron chloride and the mutated RPO iron chloride without, or the RPO without iron chloride. Through these experiments represented in this figure, ClpXP helps cell survival in high concentrations of iron, but it is not because of ClpXP's current known functions, because the mutant was not greatly impaired in comparison to the wild type, as you can tell, because there's not really a change in the figure, or in the curves. Once the determination of an additional function of ClpXP was confirmed, this allowed the authors to design an experiment to trap proteins using ClpP to determine that it traps proteins that have protein binding sites and use nucleotide-derived cofactors, 2 of which that cause high concentrations of iron anaerobically. Knowing this information overall could allow researchers to create and support their experiments for oxidative stress in aerobic conditions. This could help further the understanding and link oxidative stress to Alzheimer's.
All right, this is a little bit more comforting now, but what does current research say about the enzyme? Like, if I have it, like, do I have an inactive ClpP, or like, what's due to lead to that? Like, what's going on?
So, a molecular biology lab at Brown University, in conjunction with the molecular biology lab at the National Cancer Research Institute, just published an article on ClpXP anti-adapters. The research done demonstrated that despite a change in environment, so like a pH change or something like that in your body, ClpXP stayed resilient, so it stayed active. Their research was important because sigma S, a regulator for stress response, as I was talking about earlier, about the sigma cells, or the sigma responses as well, is regulated by ClpXP. If ClpXP became damaged or inhibited, then sigma S cannot regulate stress response, which could lead to antibiotic resistance and other processes in the body to become inefficient. Other current research is the research that Dr. Fishovitz is doing at St. Mary's College. As I told you before, there are 2 dominant domains for ClpXP, ClpP and ClpX. It is very difficult to do experiments on ClpXP without both parts being activated. So she's perfecting a way to keep ClpX activated so that experiments can be performed. She also looks at ClpXP activity in using HeLa cells, which is a human cell, in the presence of inhibitors of ClpXP and their related LON protease. Additionally, I am doing research myself on ClpXP and studying the enzymes response in high oxidative stress intracellularly, like with peroxides, as I mentioned before, and extracellularly, like with UVB light. The control is ClpXP when there's no oxidative stress present, and then I'm adding ROSs over a time course. I will then run a raster blot to see if ClpXP has been degraded over time. This could further help the understanding between oxidative stress and Alzheimer's, as I stated before.
Thank you so much for giving me all this information. It is comforting to know that researchers are researching about current diseases and one that I possibly could have.
No problem, and thank you for your time. I'll keep you informed for any other research that I know that I can tell you.
Thank you so much.